Hello, my name is Wen Wang, and I am a researcher at the University of Minnesota, Twin Cities. First, thank you for stopping by my poster and listening to my presentation. Today, I'm going to talk about my research on COVID-19. COVID-19 is highly contagious and has a high mortality rate. So far, from clinical data, we learned that COVID severity is associated with age, race, blood type, and underlying conditions such as heart problem, hypertension. But at the same time, there are also young adults without any underlying conditions still need hospital care after getting COVID. So this suggests COVID severity may be influenced by host genetic factors. There are many ongoing efforts working on the single variant front. My research on COVID-19 focused on the discovery of genetic interactions associated with COVID severity. So COVID-19 is a complex disease and genes can interact with each other to influence disease risk, which means there are variants that they don't impact disease risk individually, but when combined with other variants, they will. However, due to a lack of statistical power, genetic interactions are often very hard to be discovered by traditional pairwise approach. One important understanding of genetic interaction is that genes with similar functions are more likely to interact with each other. Our extensive study of Yeast genetic interaction network shows that genetic interactions are clustered within the same functional module or between two functional compensatory modules. So we developed a method called bridge to discover genetic interactions based on functional modules such as pathways, protein complexes. The input of bridge method is GWAS dataset and a set of pathways. Then we compute pairwise SNP-SNP interactions and construct a genetic interaction network. We then evaluate if the within pathway or between pathway structure is enriched by SNP-level interactions. We then shuffle the SNP pathway membership and sample phenotype labels to evaluate statistical significance. We applied the bridge method to 3 UK Biobank COVID-19 GWAS datasets. We used the England cohort, which has most samples for discovery, and the rest 2 cohorts as validation. We also used COVID-19 positive inpatient samples as cases, and set up 3 different controls, such as COVID negative samples, COVID positive not inpatient samples, COVID negative inpatient samples as controls. At FDR cutoff 0.05, we discovered about 300 between pathway interactions and 30 within pathway interactions. We report pathway interactions that can be replicated in both Scotland and Wales cohorts. All these interactions are linked to increased COVID-19 severity. From the name, you can tell that many of these pathways are relevant to COVID-19, such as the natural killer cell-mediated cytotoxicity, the antigen processing, and the presentation. Due to limited time, I'm not going to point out all relevant pathways, but many of them are immune response related, and their connection to COVID-19 is very obvious. Next, I want to use one example to show how we can apply some downstream analysis to gain useful information. So this is a between pathway interaction. The 2 pathways are androgen receptor and the interferon gamma signaling pathway. So here's a plot visualizing the interaction between these two pathways. The dots are SNPs mapped to the two pathways. The bars on this plot show single SNP association. So you can tell none of these SNPs will reach genome-wide significance by traditional GWAS study. The lines between these pairs of SNPs represent interactions. So this between pathway interaction has a FDR less than 0.005 in both England and the Wales cohort, so it's quite significant. We then try to identify driver SNPs from both pathways. So for example, for the androgen receptor pathway, we list the top 5 driver SNPs and the genes mapped to them. Same here for the interferon gamma signaling pathways. So driver SNPs are SNPs whose interactions are more than expected compared to its possible interactions in the genome. So interestingly, we find many driver variants located in or nearby HLA superlocus, which is quite interesting. So there's a possibility that the impact of HLA gene on COVID-19 is through interaction with other genes. We also can use genetic interaction-based polygenetic score to differentiate case and control groups. So these two box plots showing that this between pathway, if we summarize their genetic interaction scores for each individual, then we can see the case group has much higher polygenetic score than the control group. So we can use this to build a predictive model for COVID-19. In summary, our study suggests evidence for genetic interaction between common inherited variants as a modular of COVID severity. While several discovered pathways showed clear relevance to COVID-19, but most of them haven't been discovered by previous GWAS studies. So our work suggests it's important to consider genetic interaction in COVID host genetic studies. Thank you for your time and have a great day.